Hello, welcome to my lockdown lair. I'm Edward Draper and I'm here to talk to you as part of the Science Cafe at the Cut. My talk is going to try to answer a simple question. It's a simple question, but it's not a simple answer. Bones heal more easily than joints. Why is this? Why? The way in which I'm going to break this down, trying to answer this question, is I'm going to give the talk starting off talking about a bit about me, primarily to give you confidence that I know what I'm talking about, but also to try to explain the position I come to, where my viewpoint is. I want to talk about what bones and joints do, what they're made of. I do actually want to go back and start exploring the evolution of these tissues, of these structures, and to answer the question when they first appeared and why. And it's actually quite important in answering the question about healing. I'm going to talk about bones, I'm going to talk about how they heal, I'm going to talk about joints and their healing reserves. A little bit about me. I have a PhD. It is in bioengineering, and my thesis was on bone healing. I have a chartered status both as a scientist and as an engineer, and I'm quite proud of both of those. I have an academic CV in which I've published in this area in some fairly high impact journals, and I've been working for the last decade and more in the commercial world, primarily leading on the design of joint replacements, their launch, and training of surgeons with the new devices. And I'm currently working as a chief executive officer as a small startup that's actually working in this specific area, and I'll touch on that at the end. I also have a visiting professorship at UCL. But this talk's not about me. I'm doing this just to give you an indication of what my background is. So let's start off with what do bones do. I love this animation. It gives a really good sense of feel and weight, and it actually helps that to answer some of the questions, answer this question. Bones have a number of different functions. They're an important source of and store buffer of various ions. And their hollowness, you have cell factories within there, and some of the cells for other parts of the body are actually produced within the bones. But the primary function is not that. The primary function is to provide motion. The motion that allows a person or an animal to move from one place to another is referred to as locomotion. And the bones allow that while giving structure. Without bones, we would never have left the ocean. And we would have now to be carried around in a bucket if we were wanting to go in. Bones work with other aspects of the body. They work with the muscles. The muscles are the motor, the ones that provide the energy input into motion. I'm not going to be talking about those in this talk, but I am going to be talking about the other part of the body, which is essential in the understanding these structures, which is joints, and talking about their healing reserve. Bones act as spaces between the joints, and they hold the joints apart, and they have specific shapes. And you can see that if you look at skeletons of a wide range of different animals, you can see, for instance, the humerus or the femur is comparable across a vast range of different animals. They're load bearing, and they transmit loads. So if you look at the skeleton, this skeleton is able to transmit loads from the ground all the way up through the skeleton, all the way up to the skull. It's useful to see the bones as organs, but they're organs of mechanics, of biomechanics. They have a biomechanical function. What happens when they go wrong? Well, when they go wrong, they break, and you get this thing called, the technical term, medical term would be fracture, but they have an ability to heal. But let's look now at joints. Using the same animation, And now if you look at this animation, and look not so much at the bones, but look at the joints, at the knees and the elbow and things like that. And the primary function of the joint is also about motion. But without the joints, there would be no flexibility. And it's the flexibility, the power from the muscles that allow them to go through, that allows locomotion and all the other things that we do. The motions that they allow are controlled. There is a definite constraint at each of the joints that can be defined. In fact, you can buy little pamphlets that talk you through every joint of the body and gives you an indication of what the range of motion is between those. And they're quite standard. They also transfer load. They transfer load through the skeleton, and they undergo quite heavy amount of load. If you were to stand on one leg, the act of standing on one leg, the load on the hip on that leg actually exceeds 4 times body weight. And if you're walking, particularly the way the skeleton is walking, that load will be higher than that. And if you were to run or jump, it would be higher still. The other aspect of joints is that it's very important that it has low friction. And the reason for low friction is to conserve energy. The final point is that the function is all these things have to be done with no pain. If you quick look at around the screen here, pain is the only one that's not mechanical. These are mechanical structures. So like bones, joints can be considered as organs, and they're organs of biomechanics. What happens when they go wrong? Well, when these go wrong, they tend to get worse, and you tend to fall into what's referred to as joint disease. Now, before I go much further, this is a very high-level discussion of the problems of bones and joints, and the certain aspects, certain diseases that I won't be touching on. I'm not going to be touching on anything like rheumatoid arthritis or osteoporosis with bones, not because they're not important, but because I don't think we can get down into the details to discuss those, and we're going to keep them at the high level. If we look at bones, let's ask the question, what are they made of? Bones, this is a cartoon here of a femur. It's not absolutely anatomically correct, but it's not far off. And there's a few features you can see within here. The femur is described as a long bone. There are categories of bones, bones, skull, for instance, jaw, and the clavicle, the collarbone, which are not classed as long bones. But they have very similar features, and you can map them across. Looking at this then as a long bone, there's an important aspect, which is it's hollow. It's not empty, but it's hollow. And the hollowness is due to the fact that there is some very good biomechanical reasons for this. You want to have the material, the tissues, as far away as the central line as possible for it to act with the minimum mass for maximum effect. They're made up of a tissue. This is a bone tissue. And the bone tissue can come in 2 different forms. It comes in a spongy form, which tends to be at the bone ends. The spongy form is often referred to as trabecular or cancellous bone. It's not soft or pliant. It is just the fact that the bone is formed in a complex network. It also comes in the form of what's referred to as compact bone or cortical bone, and this is in the section in the center of the bone, in the diaphysis of the bone. And the tissues there are complete. There's no holes in them, except for where to let the blood supply in and out. There is another tissue associated with bones, and that's at the ends, and that's the cartilage. And I'm going to be talking about that because that cartilage is part of the joint. Bone tissue it's very important. It has supply of both blood and nerves. It requires quite a large amount of blood supply, and if the blood supply is interrupted, it will die. The tissue will die. It's referred to as avascular necrosis. It has a nerve supply, so it's able to feel feel all sorts of things, including pain. I've used the word hungry. What I mean by that is that this tissue, just to exist, uses up a huge amount of energy. The cells within bone are very metabolically active, and the bone will reduce the amount of tissue, bone tissue it has, to the minimum to be able to get away with it, what it requires to do. If you go down a level and start looking at the bone tissue, the bone tissue is normally structured with layers, a bit like tree rings or lamellae. These lamellae are actually part of the remodeling process. Bone has the extraordinary ability that even when it's intact, the bone tissue is constantly being removed and replaced. You get bone cells that eat their way through, creating little tunnels, which are then filled up behind them, and you get these filled in tunnels that I'm showing you can see on the cartoon here, these lamellae. Trapped within bone or embedded within bone are bone cells. And these bone cells that are trapped within bone are called osteocytes. And as I've mentioned, they're very metabolically active and use up quite a lot of energy. There are two other cells that are associated with bones. I'm not mentioning here because they're not involved in the function. And the two are the osteoclasts, which remove bone, and they would be the ones that would remove the bone that to create these lamellae. And then there are osteoblasts that lay bone down. And these rings are laid down by the osteoblasts. And will then embed these osteocytes, which remain within the bone. Even at this microscopic level, even at this tissue level, there is blood supply and nerves. So we've shown it at the microscopic level and down at the microscopic level. There are, within the bone tissue, small canals, canaliculi, that contain the blood supply and the nervous supply. The number of bone cells actually within the tissue is quite small, and the rest of it is referred to as a matrix. It's normally referred to as the extracellular matrix. It is that which exists that is not cells, and it is the extracellular matrix is what allows the bone to be bones. If you look at the next level down at the molecular level, there are some long molecules indicated here. 10 nanometers is a standard length of the molecule. There are 3 of them wrapped together. It is a triple helix, one better than DNA, and they form fibers. And these fibers are made from a protein. The protein is collagen. Proteins are coded for directly by the DNA, and in bone, the collagen is the collagen 1. If you look at the next level up from that, these fibril molecules will wind themselves up into larger fibrils. So you get the collagen molecule building itself up into a collagen fibril. And at that level, so this is at about 500 nanometers, you can start to see crystals that form within the fibrous matrix. The crystals don't look like this. In fact, at 500 nanometers, you're down at the wavelength of light, and you're not going to see it. You can't see anything with optical microscopes. You have to have other ways of looking at it, electron microscopes and other imaging. The crystals are not green, and the crystals aren't quite as regular as that. They're flat, but they're quite sharply crystalline. The crystals are made up of calcium salt called hydroxyapatite, and there is evidence that this hydroxyapatite has been laid down in bone for many years and will lay down in evolutionary terms, but I'll touch on that later on. So when you look at the bone as a material, as a matrix, it is fiber reinforced. And the 2 components that I'm showing here, the fibers will react tension, which allows the bone to have strength in being pulled, and The crystals provide the support to allow it to give it stiffness, to allow it to be able to expand, to react the compression. And if you can react compression and tension, you can react bending and torsion and shear. Let's move on. Let's start considering what joints are made of. Now, joints have a cavity between the bone ends, and that cavity is referred to, the technical term is the capsule, and the cartoon that I have here gives an indication of that. This is not recognizable as a particular bone, but it gives a nice cartoon flavor of what the structures are that are involved in this. The capsule is filled with fluid, the synovial fluid, and the synovial fluid is supported by a membrane. A membrane's function is biological. It is to produce the large molecules that help lubricate the joint, and they provide the nutrients. And there's quite a lot of evidence now that they're also producing some important pluripotent cells that are able to help in a number of different functions. The bearing surface, because this is a bearing that allows movement, the bearing surface is allowed not just by the synovial fluid, but by the material, by the tissue that allows that the bone ends are made from. And this tissue is cartilage. And it's calling it here articular cartilage. It's also referred to as hyaline cartilage. Hyaline meaning glass-like. It was given that by the ancient Greeks who didn't have plastic. I think nowadays we'd probably describe it as being looking like plastic, and that's certainly how it feels like when you handle fresh cartilage. Moving on to the next level, looking at the tissues, what we have here is we have an artistic impression of the tissues, sectioned up in a way that's easy to understand on the left, and on the right there is a tissue section. It's been stained. Those colours are not correct, but that is once it's been stained, and you can get some idea of the structures, and we'll talk you through those. At the bottom, there is the bone, and we talked about spongy bone, and you can see the bone here has holes in it. That's part of the sponge. So the spongy bone is quite important. There's some really good mechanical reasons as to why it needs to be spongy underneath the cartilage. But that's beyond the talk today, and I won't talk to you about that. Cartilage is on the bone end. So in this picture, it's at the top, and it's divided normally into 3 layers. There's the superficial layer, the transitional layer, and the deep layer. The main difference between these layers is that the orientation of the fibers within the tissue. At the superficial level, they're parallel to the joint surface, whereas at the deep layers, they're actually at 90 degrees to the surface and point down into the bone. You can also see that the cells are organized slightly differently. You get columns of cells at the bottom, and at the top, you get more individual cells. I'm now going to get a bit technical, because we're going to go down another level, down to the molecule. And the reason I do this will become apparent in a minute, but please bear with me. This is a cartoon of an important molecule in cartilage. It comprises of protein backbone, which is these blue circles. Proteins, as I've already said, are coded for by DNA. And shedding from these and sticking out like bristles from a brush are chains of sugars. The sugars are not properly polymerized. They're linked together with amine bonds, and you can see here there's a referent to the GAG chains. GAG stands for glycosaminoglycans, which you can break down. Glycosa means sugar, amino refers to the amine bond, glycans is also sugar, so it's sugar to sugar, which is the reason I call them sugar side chains. And this whole molecule is called a proteoglycan, and there are several of them. And we use the term proteoglycans for that. And that as a word can be broken down into meaning protein and sugar. The reason I want to talk to you about it is that when you look at this molecule, you you will see that there are many, many of these side chains. And these side chains not only have these sugars and amino links, but they also have sulfate. They have sulfur in here. And if we zoom into one of the sulfates, these sulfates are covalently bonded to the molecule. So they're not properly in solution, but they are negatively charged. And so they do have an effect on the fluid. And the fluid effect is that it's negatively charged. And so the positive ions that are within the fluids, within the body fluids surrounding these things, will respond to them. And the most common positive ion is sodium. There are many other ones as well, but it's quite a salty environment. And these sodium ions will tend to be attracted to the negative charge of the sulfate. And the consequence of this is that within solution, because these ions are in solution, is that the ion concentration is increased. It's forced to increase by this electrolytic charge. And the consequence of it being more ions in the space is that the osmotic pressure goes up. Just simple physicochemical. And the consequence is that these brush-like molecules tend to swell up. They bubble, they draw water into them. And if we go back up a little bit here, so we've got the proteoglycans. We're going back to talking about collagen. This is a different collagen. It has a lot of similarities, but it is coded for a different set of DNA. And this is collagen. This is collagen 2. And what we can see here is a mesh of collagen, and within there is some proteoglycans. And the proteoglycans are wanting to expand. They're wanting to bring water in and plump up. And resisting that is the collagen network. And the collagen pulls that in. And that creative tension actually gives this tissue its structure. And what it's doing is it's using water to act as a physical, mechanical property. Water now starts to become a structural component of this tissue. So like bone, this matrix is fiber reinforced. The tension is done by fibers. It's collagen, collagen 2. But the compression is not crystals in this case, but the compression is carried by water. It's very interesting, very, very interesting. Before I leave this a very, very important point, or two that I need to make. If you look at these pictures here, what you can see is there are no, cartilage has no blood supply, and it carries no nerves. It is avascular and it is aneural. And what that means is that it doesn't feel any pain, but it also means that any nutrients to get to these cells has to come through diffusion. So unlike bone cells, these cells are referred to as chondrocytes. Like osteocytes, these are chondrocytes, chondro meaning cartilage. The chondrocytes have very low metabolism. They don't do very much. Important point, I'll come back to that later on. Now, let's go back and do some thinking about evolution. Where do these tissues begin? And I'd like to go back a little way, 600 million years ago. That's over 500 million years ago. This is before the Cambrian explosion. And that's important. Darwin worried about the Cambrian explosion because he could see bony fossils within the rocks that were associated with the Cambrian era. But he couldn't see anything before those. And he wondered, if there was natural selection, why did these suddenly appear from nowhere? And I want to go back to that space, because what we now begin to get understanding of is that there were fossils. They're rare. They only occur in very certain parts of the world, but these are at a time where the only large organisms, multicellular organisms, had soft bodies. And let me introduce you to 3 such fossils. The first one I want to introduce you to is this one, which is Charnia. This is actually a plaster cast of China as it was discovered. It was discovered by some school children playing in Charnwood Forest in 1957, and they noticed these fossils. And it was a local geologist who came in and recognized that they were not geological, that they must have been created biologically. He thought they were plants or ferns, or he wasn't too sure what they were. Another one I'd like to introduce you to is from a different part. This is Fractophusis. This is in a large seam of these fossils that can be seen in Newfoundland, in Mistaken Point in Canada. I've never been, I've always wanted to go, possibly after lockdown. And the other one I'd like to introduce you to is Dickinsonia. Now, Dickinsonia was first discovered in Australia, in the Ediacaran Hills. And this particular image is quite interesting because it gives you three features that are associated with animals. They don't define animals, but they're associated with them. The first one is that there is a left-right symmetry. And the second one is that there is clearly front end. And the third one is there's clearly a back end. So this has the features of an animal. It is more recently that we've been able to go in and look at these fossils. And there's been some very, very clever work that's been looking at the chemistry still trapped within these fossiliferous rocks. And they're able to show that the chemistry is associated with animals and not with plants. The Dickinsonia in the Ediacaran Hills gave rise to the term Ediacaran biota, which refers to all these types of soft body fossils, and also to the era that these animals were alive in. And what we know, there's a few things that we do know. One of them is that if they're animals, they must feed, and the feeding will have been through the surface. And there are animals like that alive today and can be seen and can be studied. And they are the nearest modern animal, the sponges. It may be a surprise to some people, but sponges are considered to be part of Animalia. They are animals. They're simple animals. They are multicellular. They don't have locomotion. They don't move, but they do forcefully drive water through themselves, and they filter food through that drive of water through the surfaces. And one of the exciting things from my point of view is that what we can see with modern sponges, we can see that they have extracellular matrix. And it's not unreasonable that the extracellular matrix and the Ediacaran biota also have that as well. And if we examine the extracellular matrix, this is a paper that was published in 1975 and explored what you could see. And you can see in this image here, fibers and fibers within the matrix, and there is collagen, and there are the glycosaminoglycans. You may be able to see that there's the two letters GS. GS stands for ground substance, which is the term was used in 75. We now use the term extracellular matrix. Within that paper in 1975, this extracellular matrix was referred to as cartilaginous. And if we compare that to the cartilage cartoon diagram that I showed earlier on, there are a huge amount of similarity between that image, that microscopic image, electron microscopic image, and the diagram I've shown you there. And it is not unreasonable to think that this tissue was exactly what was around extracellularly within the Ediacaran biota. So this tissue is over 500 million years old. Not this particular one, but I mean it appeared within the ancestors 500 million years ago, over 500 million years ago. Extraordinary. The story of evolution of bone as a tissue is a little bit more complicated. It is wonderful. I would love to talk to you about it. However, I'm not going to be able to do it simply within the length of time I've got within this talk. But if we look at this particular diagram, which shows the Ediacaran era, followed by the Cambrian era, you can see that there was a sudden wiping out of the Ediacaran biota. Not completely. Sponges managed to get all the way through there, but the vast majority of them disappeared. Is why Darwin couldn't see them. And what was the difference between the Ediacaran era and the Cambrian? What happened was that bone appeared. And that's the important one, is the appearance of bone. I won't go into the detail of how that happened because it's quite complicated. But what bone allows, and it's now really quite well accepted, it not only gave protection, which probably was the original role of bone, but it also allowed improved movement, being able to swim better, being able to move, but probably a jaw, being able to open and close a mouth, and it allowed predation. And the Cambrian, the new Cambrian animals, and the innovation associated with what you can do with bone would have predated on the Ediacaran biota, which is why they were wiped out quite so quickly. Now, having talked about that, I want to come back to tissues as we are today, and it is relevant as to how do bones grow, how does cartilage grow, and it is intriguing. Let me talk to you about that. The skeleton, our skeletons first appeared when we were embryos. And they first appeared as small versions of what the bones are going to be. But initially, there's no bone tissue. Initially, they're cartilage, they're pure cartilage. They're referred to as cartilage enlarges. And what happens with these enlarges is that you get a collar of bone, which is the first formation of bone. And then with time, that the cartilage, bones grow because of cartilage. The cartilage gives them the opportunity to be able to expand out lengthwise and in width. But with time, the center of the cartilage enlarge starts to open out a little bit. And as that it triggers off, it actually triggers a signal for blood supply to come in. And the blood supply will come into the bone. And as soon as the blood supply comes in, bone can exist, and bone is then laid down and starts to pass through. Now, bone and cartilage, bone is not adding to cartilage, but it has to physically remove it and replace it and put something in, because the cartilage and the bone are genetically distinct. You get a bony front that goes through that, and remember that these bones are actually loaded, and although they're loaded, they're still able to have those functions very well. Very often with the long bones, you will get points with the ends of the bone, points of where bone islands start to grow. People will often say that children have more bones than adults. They don't. What they do is they have more of these bony growth points. These growth points then expand out until they're finally merged together as one long bone. And when they do finally merge together, the bones no longer grow. It's the point of skeletal maturity. But if you look in this cartoon, this diagram, cartilage remains. It remains at the bone ends. So that cartilage is not added as a layer. It's the other way around. Bone came in last, took over the rest of the cartilage and replaced it with bone. And if you look at that, that process of new bone coming in and replacing cartilage is very like a metaphor for the appearance of bone tissue. This process of laying down bone within cartilage is referred to as enchondral ossification. It's not the only form of ossification, but it is an important one. And we'll come back to it in a minute. If you ask the question, what happens when bones break. There is a particular form that broken bones will, and there is a healing process that they follow. I spent some time trying to work out what would be a good illustration for you for this, and I've chosen this one here. This is a bone from an Egyptian mummy. It's within the British Museum, and this bone is 2,500 years old. And what you can see here is that the bone was broken in the mid-shaft, in the mid-diaphysis, and it wasn't held properly. It's very difficult to hold on to the femur, even if you put it into a bandage or a plaster cast. It's very difficult. And the bones were malaligned. And despite this misalignment, this bone went on to heal. And it would have healed to the point where it would have caused no pain and it would have been structurally intact. This bone is clearly quite a lot shorter. So this individual would have ended up having a limp, not through pain, but through that bone length, leg length discrepancy. The bone healing process, I've already told you, it has a strong blood supply. So the first thing that happens is the blood clot appears, and the very earliest tissues appear. Earliest tissues are referred to as granulation tissues. This is highly collagen-rich fibrous material, and it doesn't carry very much weight. It's not load-bearing. Once it's been established, a new tissue can come in, a bridging tissue, and that bridging tissue in fracture healing is cartilage. When you break a bone, you recreate the evolutionary appearance of tissues, and you recreate what happened within the embryo. You first of all have cartilage, and then that is replaced with bone that comes in and takes it over. You get a bridging between the bone ends with new bone. And one of the extremely interesting things about bone healing is that it's not replaced with scar tissue. Most parts of the body, if you cut it and repair, it is repaired with scar tissue. Bone is repaired with bone. Let's now look at joints. What happens with joints? What I'm showing you here is a tissue section. So it's a very thin section cut through a joint. These colors are not natural. They've been stained. The stain in this case, well, it was more than one stain, but the stain of importance is saffron O. This allows us to see where the proteoglycans exist. Now, as I've told you, the proteoglycans is important in terms of in trapping the water and allowing this tissue to function as a tissue. As the disease progresses, what you can see is that proteoglycans start to disappear, and you get this front of proteoglycan loss. And after a while, as it loses that tissue, the proteoglycans are lost, it loses the water, and it becomes very fibrillar, it starts to break up. As the proteoglycans are disappearing further, you get this fibrillation and the tissue breaks up. And eventually you get to the point where the proteoglycan are almost completely gone. The important thing is that as it begins to lose the proteoglycans, it is no longer able to function. It's no longer able to carry the loads that it needs for its primary function. And the consequence is that as it begins to break down, it breaks down fast, and you end up with this degenerative cascade. This is often referred to as joint disease or degenerative joint disease, and it leads to osteoarthritis. I tried to, again, come up with a way of illustrating this, and I found this picture. This is a picture of a knee. And you can see here it's quite badly affected by joint disease. This is work that was done on a study of Native Americans. And this is a photograph from the Smithsonian Institute. And this particular bone is 6,000 years old. And what you can see here is in one of the compartments, the femur, which is on the left, and the tibia, which is on the right, have been worn down in one compartment, down to bone on bone. This would be very painful. But this individual has clearly carried on. And because bone has nerves, when you go bone on bone, you get that pain, you get that joint pain. And the cartilage, which is annual, has been lost. So you get that. The current standard treatment, gold standard for this type of disease at this state, would be to remove this joint, is to cut out this joint and put in metal and plastic, or metal and ceramic, and do a replacement. I've suggested a total joint replacement, but it is possible to replace just that compartment, just that part of the knee there that is affected. Joint replacement doesn't return to full health. What it does, it's a replacement. It is a prosthesis. So in summary, where are we? Well, joints, the most important part of the joint is the bearing surface, which is the cartilage. The cartilage forms that bearing surface. We know that it is a very ancient type of tissue. It's associated with very poor healing reserve. And when it is damaged, when it starts to try to heal, it is no longer able to withstand the loads that it's been carried, and you get a degenerative cascade leading to joint disease. Bones, on the other hand, which act as spacers, have excellent blood supply, good nerve supply, and they have excellent healing reserves. And they will heal, even if they're not aligned. So part of the process of treating a broken bone is to make sure you're holding them together in the right way, in the right alignment. And hopefully that explains why bones heal better than joints. But before I leave, the point about cartilage is there is still hope. And there are some really exciting new technologies that are being developed to help treat cartilage, to regrow cartilage, and to help the bone healing process. And these are being developed at the moment. In fact, my company is part of that work that's ongoing. But I'm not going to talk to you about that. Possibly another talk, and hopefully with me in person, talking to you in the cut. Thank you very much.